All right, so for this video, we're going to put our focus on the typical male reproductive system. Again, I'm saying that over and over again because it really is important that we recognize that there are some differences that can exist in development when it comes to reproductive system, but we don't have the time or the space to discuss all of those ways things can change, and so we're going to focus on the typical male reproductive system development and physiological processes. So I'm not going to keep saying typical male reproductive systems of this video, just know that's what I mean as we go through it. So we're going to talk about male reproductive structures. We're going to talk about the Sertoli cells and the Leydig cells in a little bit more detail. We'll talk about spermatogenesis and the different ducts we find, the different accessory glands we find, innervation patterns when it comes to male sexual response, and then hormonal regulation when it comes to the male reproductive system as well. So, if we're looking at the male reproductive system, we are looking at gonads, ducts, accessory glands, and supporting structures. When it comes to gonads, we're looking at the testes. This is where sperm will be produced. The ducts, like the epididymis, vas deferens, ejaculatory duct, and urethra. The accessory glands, like the seminal vesicles, prostate gland, bulbourethral glands, and then supporting structures like the penis, the scrotum, right, and the muscles and the nitty-gritty of the scrotum that will go with it, like the cremaster muscles, that will help with the temperature control, right, the thermostat, basically, of the testes. So let's go ahead and talk about the testes a little bit more. Again, inside the testes, we have what are called seminiferous tubules. This is where we are producing sperm. So inside of these seminiferous tubules, we have Sertoli cells and Leydig cells. The Sertoli cells, these guys that we see here, right, in kind of like this pink color, they are going to form the blood-testis barrier. This is going to help prevent the person's own body from attacking the sperm cells, because the sperm cells are different, right? They're not going to be 100% genetically identical. And so we have these tight barriers like this, right, that are formed that are going to help prevent the immune system of that person from destroying the sperm. They're also going to help with the process of spermatogenesis. They're going to produce inhibin, which is going to reduce spermatogenesis. So if we already have enough sperm produced, inhibin will basically make its way up to the anterior pituitary and the hypothalamus to turn down the hormonal control from those organs to the gonads. The Leydig cells, right, are the ones like these guys up here. These guys are the ones found between the seminiferous tubules, and they're going to produce testosterone. And that's also going to assist with spermatogenesis. It's going to help promote that. And so if we look at how this process of spermatogenesis goes, it tends to follow what you've already learned when it comes to meiosis. But we're going to talk about some of the nitty-gritty, because we're going to start, our parent cell is going to be the spermatogonia. These are the stem cells for sperm. They're diploid, and they will go through meiosis to produce those haploid sperms. So we start with spermatogonium, this guy here. We go through mitosis to produce some primary spermatocytes, or still 2N. We go through meiosis 1 to get a secondary spermatocyte, which means we're already down to N, and then we produce a spermatid, which is at the end of meiosis 2. And then finally, in our maturation process, or at least to get a little bit more mature, we produce a spermatozoa, or a sperm cell. When we release the sperm from these Sertoli cells, we call it spermiation. And so we can really divide this process into 3 different spots. We have proliferation, which is mitosis, and differentiation. This is where we're producing a spermatocyte from a spermatogonia. Meiosis, which is the second part, where we're going to go through to produce our secondary spermatocyte and spermatids, where we now go from diploid to haploid. And then spermiogenesis, this is where we are going to go through differentiation to go from just our plain old spermatids to a full-on sperm cell that has the head and the body and the tail, right? That they're going to have very specific structures to that sperm, right? Things like the acrosome, things like the the massive amount of mitochondria that are packed in there, the flagellum, right? Those are all going to come forward in spermiogenesis. And so here is the different pieces of a sperm cell. So this is the final product. This is what's going to try to get into an egg or an oocyte. And so we have the head, which has the nucleus. So that head contains the nucleus. It also has an acrosome over the top. The acrosome is going to have digestive enzymes in it to help with getting into the egg. So these digestive enzymes will eat away at the tough exterior of an oocyte or of an egg to be able to push that DNA inside. The neck is going to have centrioles to help make up the tail. The middle piece is where we find the mitochondria, where these mitochondria are going to support basically what's called flagellation, or the movement that's required, because these guys have to swim and you need energy to swim. So mitochondria have got to be there in high numbers. And then the tail is the end piece, and this is going to be used to move that sperm. So it's used to swim. When we look at some of the key anatomy, I'm going to go through some of this stuff pretty quickly because you should have the anatomy stuff at least somewhat familiar. Basically, where we have sperm and the fluid that goes through the semen from seminiferous tubules to the straight tubules, and then to a network of ducts that we call the rete testis. From there we have efferent ducts, remember efferent exits, that are going to carry sperm out of the testes to the epididymis. The epididymis is where we can store sperm for maturation because they're not quite ready yet, as well as break down any sperm that are unused and resorb all the useful bits. Sperm can stay there for about 10 to 15 days. After that, they are no longer really useful or like viable, and so we'll break those down and produce more to replace them. From the epididymis, from sperm, we'll move into the ductus deferens, sometimes called the vas deferens. I personally know it best as vas deferens, so it's probably the term I will use. From there, out of the ejaculatory duct and into the urethra, we're using smooth muscle for this during ejaculation. And so this is why you hear of a vasectomy. What is a vasectomy? A vasectomy is a sterilization process in people with penises and testes where we cut and we basically sever the vas deferens to prevent sperm from being able to get up towards the accessory glands. So if someone has a vasectomy, they will still produce sperm, they'll still produce testosterone, they'll still produce semen, all of that. The only difference is the sperm have nowhere to go. And so because the sperm have nowhere to go, they just basically are produced and break back down again. This person who has a vasectomy cannot impregnate somebody. There's no sperm in the semen. The semen would basically be empty. And that's why you hear people talk about it in a crude way when they say firing blanks, because the person will still be able to ejaculate. They will still produce semen. There's just no sperm in the semen. So for anyone who's ever thought about it but has been worried about it, quote, taking away masculinity, a vasectomy does not do anything of the sort. That person still produces sperm, they still have testosterone, they still have semen, all of that. The only difference is the sperm can't actually leave the testes area very well at all. So let's talk about those accessory glands. Since we're talking about the production of semen and that someone with a vasectomy can still produce it, where is the semen coming from if it's not coming from the testes? So it's going to come from these three different glands, the seminal vesicles, the prostate, and the bulbourethral gland. The seminal vesicles, these guys are producing an alkaline, so a basic fluid that has some sugars in it. So this is going to basically provide energy for sperm. So all that fructose and whatnot is used to support the sperm and other mitochondria. The prostate, which is going to have a slightly acidic fluid with some citric acid, some proteolytic enzymes, some acid phosphatase, and seminal plasmin. Right, this is going to help with some of that for lubrication. And then the bulbourethral glands, this is going to be very alkaline, that's produced during sexual arousal to neutralize the acidic environment that we find inside the vagina, because the vagina is relatively acidic in comparison to other body parts, and so it's kind of a hostile environment for any other types of cells. And so the bulbourethral gland will put things in the semen that will neutralize that acid, right, as well as provide some lubrication as well. So semen, the seminal fluid, is a mix of spermatozoa and all of these sex gland secretions for the spermatozoa to be transported in. Again, it provides the nutrients. It's going to neutralize the acidic environment, both in the male urethra, because remember the male urethra will also provide a space for urination, and urine is a bit more acidic, as well as the acidic female vagina. It also has an antibiotic in it, the seminal plasmin, and prostatic enzymes. This is going to help with the movement through the uterine cervix, because that cervix, right, it has to get up to that area and then get through the cervix to get into the uterus. When we look at seminal fluids breakdown, we see that only about 3% of it's actually sperm. Most of it is seminal fluid, with about a quarter coming from the prostate and a small percent coming from the bulbourethral fluid. And so you can see how if someone were to have a vasectomy, they're not losing much in terms of the content of semen. Most of the semen is coming from these other accessory glands, and that's still all going to be fully intact. The average ejaculate is about 2.5 to 5 milliliters and has about 50 to 150 million sperm per milliliter. It is slightly basic because it's a 7.2 to 7.7. And so again, this is to neutralize any sort of low pH or acidic pH from the urethra in the male while it's traveling through, as well as the most definitely acidic environment we find in the vagina. So now we got to get into some of the physiology. This is what I'm going to worry about more, because this is going to be, generally speaking, new information. But we get into the erection, and how does the erection come about, and how does ejaculation happen? An erection is just the preparation of that tissue for sexual intercourse. It's stiffening of that tissue so that it can easily be used in penis to vagina intercourse. This is going to be brought about by the parasympathetic nervous system. So basically the parasympathetic nervous system is going to cause vasodilation in the arterioles in the erectile tissues, namely the cavernosa and the spongiosum. So it's going to be these guys here. So this here in this kind of lightish pink, and this here in this orangey pink, are going to basically be flooded with blood, right? And so this is what, all this blood is kind of getting into this tissue, this spongy tissue, which is going to dilate these blood sinuses, causing what's called tumescence, which means swelling, right? So this swelling of these tissues, because they are engorged with blood, is what leads to the erection. Now, this will be maintained during, generally speaking, will be maintained during sexual intercourse, and then towards the end, we will experience ejaculation, which is that forceful release of semen. This is coming from the sympathetic nervous system. So the muscles that we have, the bulbous spongiosus, the perineal, and the ischiocavernosus muscles are going to contract, and when they do this, this is going to force semen from the vas deferens and the urethra. The accessory glands fluid is going to come along the way too, and it's going to all come out, right? It's going to be pushed out through the urethra. When it comes to hormonal regulation, hormones are deeply integrated. We kind of barely talked about these guys when we talked about endocrine, and of course we got to come back to it because there's overlap. I already mentioned in a previous video or earlier on about the hypothalamus and the pituitary gland playing a role, because gonadotropin releasing hormone is going to be released by the hypothalamus, received by the anterior pituitary, and then the anterior pituitary in response will release luteinizing hormone, or LH, and follicle stimulating hormone called FSH. LH is going to stimulate Leydig cells to produce testosterone. FSH will stimulate spermatogenesis. From there, the testosterone that we produce is going to help control growth, development, the sex organ function, bone growth, protein anabolism, so breaking down proteins and utilizing those, building up proteins, sperm maturation, and whatnot. And of course, when we have enough testosterone, it will cause negative feedback, meaning once we reach a certain level of testosterone, it will inhibit the production of the gonadotropin releasing hormone, luteinizing and follicle stimulating hormone. Inhibin is produced by the Sertoli cells. This is going to inhibit FSH, right? So it's going to inhibit the anterior pituitary from talking to the gonads. And so if we take a look here at the picture here, right, we kind of see how all this plays out and how they are aligned. So you see the solid green lines are going to be promoting things. So they're promoting something to happen. They're stimulating something to happen. And the dotted or the dashed red lines are going to be inhibitory pathways.